Hello students of class 12. Welcome back once again to your biology class. So in our last class we have completed the chapter of molecular basis of inheritance. I think you have been well versed with that chapter. So let us start a new lesson today.
Our lesson for today is biotechnology, principles and processes. So let us start. First you have to know what is biotechnology. Biotechnology deals with different types of microorganisms, plants or animal cells or their enzymes to produce products and processes useful to humans. Carl Erke first coined the term biotechnology. Now, what are the principles of biotechnology? Basically, biotechnology deals with, you can see in the screen, micro-mediated process, in vitro fertilization, synthesis and using of a gene, preparation of DNA vaccine and correcting a defective gene. What are the principles of biotechnology? The two core techniques that developed modern biotechnology are first is genetic engineering and second is bioprocess engineering. Genetic engineering is a modification of chemical nature of DNA and RNA and their introduction into another host organism to change the phenotypic character of the host. Okay, and bioprocess engineering, it is the maintenance of sterile ambience in chemical engineering processes for growing desired microbe or prokaryotic cell in large quantities for the manufacture of antibiotics, vaccines, enzymes, etc. Now, what are the basic steps in genetic engineering? First, identification of DNA with desirable gene. Second, introduction of the DNA into host to form recombinant DNA or we can say rDNA. Third is maintenance of DNA in host and gene cloning. And finally is the gene transfer. And the steps which are carried out in constructing the first recombinant DNA is first the gene encoding antibiotic resistance in the native plasmid of Salmonella was identified. And plasmid is an autonomously replicating circular extrachromosomal DNA. Okay, and second step, what is done here is the desired gene or the desired DNA is cut at specific locations by restriction enzymes. And that cut DNA is linked to plasmid DNA and then these are transferred to Escherichia coli for gene multiplication. Understood? These are the three steps which are carried out in constructing the first recombinant DNA or we can say as rDNA. Now, what are the tools of recombinant DNA technology? The key tools which are required for the recombinant DNA technology are restriction enzymes, polymerase enzymes, ligase, vectors, and first or the utmost requirement is the host organism or the cell. Okay, these are the tools of recombinant DNA technology. The restriction enzymes are also known as molecular scissors, and these are basically responsible for cutting the DNA. Where they are present? They are present in bacteria to provide a type of defense mechanism which is known as restriction modification system. The first restriction endonuclease, HindII, was isolated from Haemophilia influenzae bacterium and it was used to cut the DNA molecules at a particular point by recognizing a specific sequence of 6 base pairs which is known as recognition sequence. Clear? This is the restriction enzymes and they belong to a class of enzymes which is known as nuclease. Now, how these restriction enzymes are named? The naming of the restriction enzyme. The first letter is derived from the genus name, and the next two letters will be derived from the species name of the prokaryotic cell from where the enzyme is extracted. Okay, and the Roman numbers following the name, they show the order in which the enzymes were isolated from the bacterial strain. Okay, and if you see here, you can see in the screen echo R1. This is derived from Escherichia coli RY13. Hint 2 is extracted or it is derived from Haemophilus influenzae. BAM H1 from Bacillus species. So restriction enzymes, they belong to a class of enzyme which is known as nuclease, and these are basically of two types. One is exonuclease, another is the endonuclease. Exo means outside and endo means inside. So exonuclease, they cut the DNA at the ends, okay, at the last end point. And endonuclease, what they do, they cut the DNA at specific position. Clear? This is the main difference between these two. And the recognition sequence of the endonuclease are palindromic. Palindromic means the sequence of the base pairs will be read the same on both the DNA strands where the orientation of reading is kept the same. Okay, so if you read at GAATTC, and palindromic will be CTTAAG. So this is known as the palindromic sequence. Clear? Now, what is the mechanism of action of the endonuclease? Every endonuclease, basically, they inspect the entire DNA sequence for the palindromic recognition sequence. When they find the palindrome, the endonuclease will bind to that specific DNA and it will cut the opposite strands of the DNA in the sugar phosphate backbone, which is situated a little away from the center of the palindromic site, but between the same base on both the strands. Clear? And the sticky end, what they will do, basically you have to know what the sticky ends. Let me tell you first. The formation, due to this, the formation of the single stranded overhanging stretches will be formed at each end of the strand, which is known as the sticky ends. In genetic engineering, basically the DNA from the different sources are cut with the same restriction enzyme so that both the DNA fragments will have same kind of sticky ends. Understood? Now let us come to cloning vectors. What are the cloning vectors? The vectors are the DNA molecules that can carry a foreign DNA segment into the host cell. Vectors, it may be plasmid or it may be bacteriophage. Plasmid are autonomously replicating circular extrachromosomal DNA. Okay, and bacteriophage are the viruses which infect the bacteria. Now, copy number, there is a term. What is that? Copy number, you can say it can be defined as the number of copies of vectors which is present in a cell. And the best known vector is the plasmid vector. Okay, and PBR 322 is the first artificial cloning vector which was developed from Escherichia coli plasmid. Understood? Now, what are the features required for cloning into a vector? First is the origin of replication, or we say as ORI. Second is selectable marker. Third is cloning sites. And number 4, vectors for cloning genes in plants and animals. Okay, so first, origin of replication. This is a DNA sequence. The origin of replication is a DNA sequence that is responsible for initiating the process of replication. Any piece of DNA, when it is linked to this sequence, it can replicate within the host cell. origin of replication, it also controls the copy numbers of the linked DNA. Okay, and second is selectable marker. What you are studying here is these are the following features which are required to facilitate the cloning of a vector. Okay, second is selectable marker. First we have discussed about origin of replication. Number two, selectable marker. Basically, this selectable marker, it helps to select the host cell which contain the vector or transformants and eliminate the non-transformants. Now, what is transformation? Transformation is defined as the procedure by which a piece of DNA is introduced into a bacterial host. And genes which encode the resistance to antibiotics, like you can say ampicillin, tetracycline, these are useful markers, useful selectable markers for Escherichia coli. The normal Escherichia coli, basically they do not carry the resistance against any of these antibodies. But these antibodies like ampicillin, these are used as selectable markers only. Okay, next we come to the cloning sites. To link the alien DNA, the vectors require very few recognition site for the restriction enzyme. More than one restriction site, or more than we can say more than one recognition site within the vector can complicate the gene coding process because it will generate several fragments. And the ligation of the foreign DNA or the ligation of the alien DNA, it can be carried out at a restriction site which is present in one of the two antibiotic resistance gene. Okay, you have to practice this diagram of E. coli cloning vector PBR322 which shows the restriction site. Okay, you have to practice this diagram along with labeling its positions. Now the vectors for cloning, this is the fourth point, fourth feature which is required to facilitate the cloning into a vector is the vectors for cloning genes in plants and animals. Various vectors or several vectors are used for cloning the genes in the plants and the animals. In plants, the tumor inducing plasmid, TI we can say, of Agrobacterium is used as a cloning vector. Agrobacterium tumefaciens is a pathogen of several dicot plants. What does it do? It delivers a piece of DNA known as tDNA in the TI plasmid, which transforms the normal plant cells into tumor cells and produce the chemicals against the pathogen. Okay. And retrovirus, adenovirus, and we can say papillomavirus, these are now used as cloning vectors in animals. Why? Because they have the ability to transform the normal cells into cancerous cells. Clear? And this selection of this recombinant form, it can be done in different methods. First method applied can be inactivation of antibiotics, and another process by use of insertional inactivation. How the selection of the recombinants formed, it can be done by this method. First we will discuss about the inactivation of enzymes. If a foreign DNA ligase, if any type of foreign DNA ligates at the BAM-H1 site of tetracycline resistance gene in a vector, if you say PBR322, then the recombinant plasmid will lose the tetracycline resistance due to the insertion of the foreign DNA. And it can also be selected out from the non-recombinant ones by plating the transformants on ampicillin containing medium. And these transformants basically we say which cause the transformation, growing on ampicillin, they will contain a medium They will contain a medium, and then they will be transferred onto a medium which contains tetracycline. And these recombinants can now grow in ampicillin, which ampicillin containing medium, but not on that containing tetracycline, where non-recombinants can grow on the medium containing both the antibiotics, and thus recombinants are selected. Second process which can be used is insertional inactivation. Sorry. On the basis of the color production in the presence of chromogenic substance, chromo means color, so which cause uh, color, which can form different type of color. The recombinants and non-recombinants can be differentiated based on the presence of the chromogenic substance in them. Here what happens? The recombinant DNA is inserted within the coding sequence of an enzyme beta-galactosidase, which will result into inactivation of the enzyme. And the bacterial colonies will have inserted plasmid, which will show no coloration, while those without the inserted plasmids will have blue color. Okay, and the competent host for transformation within the recombinant DNA, the DNA is a hydrophilic molecule and it cannot pass through the cell membrane. We all know this. So that bacteria should be made competent to accept the DNA molecules. And what is competency? It is basically the ability of a cell to take up any type of foreign DNA. And the cell is made up competent by different methods. First, you can use chemical method and physical method. In chemical method, the cell is treated with specific concentration of divalent cation like calcium. Why? To increase the pore size of the cell wall. And the cells are incubated with recombinant DNA on ice, which is followed by placing them briefly at 42 degrees Celsius and then putting them back into ice. This process is known as the heat shock treatment. And now this bacteria will be able to take up the recombinant DNA. Understood? And in physical method, we use the microinjection method, or we use the biolistics or gene gun method. In microinjection method, what happens? The recombinant DNA is directly injected into the nucleus of an animal cell. And in biolistics or gene gun method, what happens? The cells are bombarded with high velocity microparticles of gold or tungsten, which is coated with DNA in plants. Okay, and discerned pathogen vectors are also used to transfer the rDNA. So different processes can be used. Next process of recombinant DNA technology. Recombinant DNA technology, it involves different steps. First, isolation of the genetic material or DNA. One thing I want to say here, I think I should have told it beforehand, that is about the gel electrophoresis. I'm coming back to it later on. The separation and isolation of the DNA fragments is done by the process of gel electrophoresis. What is gel electrophoresis? It is a technique for separating the DNA fragments based on their size. Okay, first what happens? The sample DNA, it is cut into fragments by using restriction endonuclease. Then the DNA fragments, it becomes negatively charged and it can be separated by forcing them to move towards the anode under an electric field through a medium or matrix. Commonly used matrix here used is agarose, which is a natural linear polymer of D-galactose and 3,6 anhydro L-galactose, which is extracted from seaweeds. Okay, and the DNA fragments, it separate out or it resolves according to their size. Why? Because they have the saving property of, because the agarose gel, they have the saving property. So the smaller fragments size and far the smaller will be the fragment size, the it will move farther away. Okay, and the separated bands of DNA, it is visualized by staining it with ethidium bromide followed by exposure to UV radiations and the separated band of DNA are cut out and extracted from the gel piece. This step is called elution. And this purified DNA fragment, these are now used to form recombinant DNA and it can be joined along with the cloning vectors. Okay. Now the process of recombinant DNA technology, it has different steps. First, isolation of DNA. Isolation means separation, you know. RNA is removed here in this process by treatment with ribonuclease and protein, and removed by the treatment with protease. After several treatments, the purified DNA is precipitated by adding chilled ethanol and the bacterial, plant cell or the animal cell which is used is broken down by the enzyme to release the DNA along with RNA, protein, polysaccharide and lipids. Okay. And the bacterial cell is treated with the enzyme lysozymes. Plant cells are treated with the enzyme cellulase and fungal cells are treated with the enzyme chitinase. Clear? This is isolation of the genetic material. You can see here in this slide. Next is cutting of DNA at specific locations. Now the DNA it should be cut by using the restriction enzyme. The purified DNA is incubated with the specific restriction enzyme at conditions optimum for the enzyme to act. Okay, and after that we know that DNA is negatively charged, and by this process of agarose gel electrophoresis, these DNA fragments can be easily separated. And it can be seen as bright orange color bands when they are stained with ethidium bromide and exposed to ultraviolet radiations. You can see in this slide how it looks like. Okay, next is isolation of desired DNA fragments. I told it, okay. By this agarose gel electrophoresis, this is since the DNA is negatively charged, it moves towards the positive electrode or anode, and in this process the DNA fragments will separate out based on their sizes. Okay. Next is amplification of the gene of interest using PCR. PCR means polymerase chain reaction. It is done in which amplification of specific DNA sequence is carried out in vitro. And this technique has some requirements. What are the requirements for PCR? Polymerase chain reaction technique, DNA template, primers and enzymes. These are required. The requirements for performing the process of PCR. DNA template, the double-stranded DNA, it needs to be amplified, and the primers are the small chemically synthesized oligonucleotides of about 10 to 18 nucleotides, which is complementary to a region of template DNA. Two commonly used enzymes used as are Taq polymerase, which is isolated from thermophilic bacterium Thermus aquaticus, and Vent polymerase, it is isolated from Thermococcus litoralis. Okay, and now PCR is carried out by different steps. First step is denaturation, second step is annealing, third step is extension. Okay, these are the three steps in which PCR or polymerase chain reaction is carried out. First step, when it comes to the question, you have to write about PCR, few sentences you have to write about PCR, and then you have to write about the steps. You have to write in points. Basically it will carry more marks for you. You give the points. Number one, denaturation. First you write a short about PCR, then the steps, about steps you write denaturation, write 2 sentences, annealing, write 2 sentences, extension, write 2 sentences. In this way you follow. Now denaturation, let us discuss about this point. The double-stranded DNA is denatured by subjecting it to a high temperature of 95 degrees Celsius for 15 seconds. And each separated single strand will now act as template for the synthesis of DNA. Second step is annealing. 2 steps of primers are added, which anneal to the 3' end of each separated strand, and primers will act as initiators of replication. Okay. Third step is the extension. DNA polymerase will extend the primers by adding nucleotides complementary to a template which is provided in the reaction. And a thermostable DNA polymerase, which I told, Taq polymerase, TAQ, we say TAQ polymerase is used in the reaction, which can tolerate the high temperature of the reaction. Okay, and these steps are repeated many times, and several copies of desired DNA is extracted after that. Okay, in the process of next DNA technology, the ligation of the DNA fragment into a vector. The vector DNA and the source DNA, these are cut with the same endonuclease. Why? Because the sticky ends should be obtained at both the corners, otherwise it cannot be ligated. And these are then ligated by mixing vector DNA, gene of interest and enzyme DNA ligase to form a recombinant DNA. Understood? And after that, next step done is insertion of the recombinant DNA into the host cell or organism. Now, introduction of the ligated DNA into the recipient cell, it occurs by several methods before which the recipient cell is made competent to receive the DNA. If the recombinant DNA carrying antibiotic resistance like ampicillin, that is transferred into E. coli cells, the host cell is transformed into ampicillin resistant cell. And the ampicillin resistant cell in this case is called the selectable marker. And on growing the transformed cells on agar plate, which contains ampicillin, only the transformants will grow and the others will die. Yes? After that, the culturing of the host cell is done. How? The transformed host cell are growing in appropriate nutrient medium at optimal conditions, and the DNA it gets multiplied and it expresses itself to form the desired product. The next step which comes is the extraction of the desired gene product. What happens here when a protein encoding gene, we all know, it is expressed in a heterologous host that is known as recombinant protein. And the cells having genes of interest, it can be grown on a small scale or on a large scale. If it is grown on a small scale means the requirement is less, the cells is grown on cultures in laboratory, and then the expressed protein is extracted and purified by different separation techniques. And on large scale production, the cells are grown in continuous culture system in which a fresh medium is added from one side to maintain the cells in exponential growth phase, and the desired protein is collected from the other side. And on large scale method, what happens? The larger biomass is produced and it leads to high yield. Next is the downstream processing. All the process in which a product is subjected to before adding it to the market is a finished product. This is known as downstream processing. It includes different steps, like first what is done, the separation of the product from the reactor. Second is purification of the product. Third, what is done, formulation of the product with the suitable preservatives. And finally, the quality control testing is done, and clinical trials are also done in case of drugs. So these are the 9 steps which are used, or steps which are followed in the process of recombinant DNA technology. So let us summarize the recombinant DNA technology. What are the steps? First, isolation of DNA. Number 2, fragmentation of DNA by restriction endonuclease. Third, isolation of a desired DNA fragment. Fourth, amplification of the gene of interest. Fifth, ligation of the DNA fragment into a vector. Sixth, insertion of recombinant DNA into a host. Seventh, culturing the host cell on a suitable medium at a large scale. Eighth, extraction of the desired gene product. And number 9, finally, the downstream processing of the products as finished product which is ready for marketing. Okay, these are the steps, or sorry, these are the processes, we can say, processes of recombinant DNA technology. Now, what are the goals of recombinant DNA technology? Recombinant DNA technology is followed, it should have some goals. What are those? First, to isolate and characterize a gene. Next, to make desired alterations in one or more isolated gene. Next, to return altered gene to living cells, artificially synthesize new genes, alternating the genome of an organism, understand the hereditary diseases and their cure, and improve human genome. These are the goals of recombinant DNA technology. Next, bioreactors. What are bioreactors? Bioreactors are the vessels of large volumes in which the raw materials are biologically converted into specific products. Okay, and these bioreactors provide all the optimal conditions for achieving the desired product by providing the optimal growth conditions like temperature, pH, substrate, salts, vitamins, oxygen, etc. Stirred tank bioreactors are commonly used bioreactors, and these are cylindrical with curved base to facilitate the proper mixing of the contents. So here you have to practice the diagram of both the reactors. Okay, and The bioreactor has the the components. Basically, what are the components or parts of a bioreactor? An agitator system is there, an oxygen delivery system, foam control system, temperature control system, pH control system, and sampling ports to withdraw the cultures periodically. Okay, so you have to draw and level the bioreactors, and you have to write the components of the bioreactor. And the stirrer, what is the function of the stirrer? It mixes the contents and makes oxygen available throughout the bioreactor. And apart from this, sparge stirred tank reactor is a stirred type reactor in which air is bubbled out, and the gas entrainment is there, and it increases the surface area for the transfer of the oxygen. Okay, and here the bubbles dramatically increase the oxygen transfer area. Clear? So this is all about the chapter. Read properly. Thank you so much.